Okay. First one I got parts wrong on was question 6 FRQ. Question 6 was the FRQ with the meiotic appearance of root tip cells. Part A read, describe the structural features of the two ends of a single nucleotide chain. Explain how the addition of a chemical that disrupts hydrogen bonds will alter the structure of DNA isolated from onion root tip cells. I said nucleotide chains read 5' to 3'. They have a phosphate group and 5 carbon sugar near the 5' and a nitrogenous base near the 3'. Breaking hydrogen bonds in DNA structure would disrupt the bonding of a strand to another strand. This is the bond that connects two nitrogenous bases. The bonds inside the single strands not connecting nitrogenous bases are phosphodiester bonds. My description did not include the hydroxyl group, and also it did not explain that it causes that the hydrogen bonds when it breaks cause two separate backbones to break. So I would say nucleotide chains read 5' to 3'. They have phosphate group and 5 carbon sugar near the 5' end and nitrogenous base near the 3'. Breaking hydrogen bonds in the DNA structure would disrupt that the hydroxyl group is by the 5' end. Then I would say that breaking hydrogen bonds in the DNA structure would disrupt the bonding of one strand to another strand to separate backbones that are separated. In part B, it says, based on table 1, identify an independent variable in the scientist's investigation. Justify the use of mutagen X in one of the treatment groups. Justify the scientists examine the same number of cells in each treatment group. I said, identify variable is a solution added to the bulbs. Mutagen X should be used because it can be used to predict the effects of normal mutagens. Use the same number of cells in each treatment group adds control to that variable. If different number of cells are used, that would be an extraneous variable. I got the independent variable correct. I had to say that mutagen X was a positive control, and I said it was a constant one. And I said that it was a control when it was a constant. So correct answer would be the independent variable is a solution added to the bulbs. Mutagen X should be used because if it can be used to break the effects of environmental mutagens, because it is a positive control, and then the same number of cells was a constant. In part C, it's based on the data in table 1 and figure 1. Describe the general relationship between the meiotic index and the percent. I said there appears to be no relationship because I was sort of rushing this question. The correct answer is that as the meiotic index increases, the percent number of cells with chromosomal or meiotic abnormalities decreases. Finally, part D. The scientists then analyzed the DNA from root tip cells of Acepa bulbs that are maintained in water from location A for 1 week and are transferred to fresh water for 1 week. Assuming mutations do not result in cell death, use the data in table 1 and figure 1 to predict how the amount of cells mutation means bulbs we compare with the amount of cells expected to have mutations in bulbs maintained in fresh water and bulbs maintained in water from location A for the full 2-week period. Based on the data, provide reasoning to justify your prediction. Explain why the meiotic index of cells in the root tip might be altered by a mutation that affects the meiotic spindle. I said less mutation present in location A in freshwater cells is assumed from the table. The meiotic index is altered because meiotic spindle is altered. I said, anyway, I need to include that a justification that water from location A induces more mutations than does fresh water. So the longer the bulbs are in location A, water, the more mutations you have. And that if mutation is in a gene encoding a spindle protein, then chromosomes will not align to separate correctly, and the cells may not complete cell division. Okay. The next one that I got wrong was question 18. Question 18 dealt with Stickleback. question said, which of the following best describes how sticklebacks in the same population with identical copies of the PITX1 gene can still show phenotypic variation? I said answer D. The subcellular location of the PITX1 gene changes when individuals move to an environment. This is wrong because this just doesn't make sense that the gene would change the location when it was in the environment. I think I didn't read it well enough. Part A said PITX1 genes carried in different chromosomes of different individuals. This doesn't make sense that carry on the same chromosome in every individual. Part C, the genetic code of the PITX1 gene is translated directly in males and females, or differently in males and females. This is wrong. It's not a X-linked chromosomal. And question B, the right part said expression of the PITX1 gene is affected by mutations at other genetic loci. This makes sense because the loci might still affect that because of the phenotypic expression. 19, I got wrong. It said gibberellic acid stimulates the cells of germinating grass seeds to produce mRNA molecules that code for hydrolytic enzymes. In this case, the role of gibberellic acid can best be described as that of, I said E, an activator of translation. It says because mRNA is involved in translation, but it doesn't make sense because this is not that part of the mRNA, like, usage thing. Question part B said a simulator of hydrolytic secretion. This doesn't make sense because it's not part of the secretion, it just codes for the enzymes. A simulator of DNA replication. This mRNA is not involved in DNA replication. An allosteric activator of hydrolase. This does not make sense because the enzymes only make them to activate them. A regulator of gene activity does make sense because it is regulating the gene activity of the hydrolytic enzyme streams. Question 20 was calculate the minimum number of nucleotides required in the coding region of the LDS2B mRNA molecule to produce and terminate the 565 amino acid and polypeptide. Provide two reasons the number of nucleotides in mature mRNA may differ from the number of base pairs in the gene. I got the point for 1695. But I got the part for 1695 because 565 amino acids times the nucleotides. But my reasoning was off. I said one reason is the mRNA will lose introns, but the genome structure obviously will not. Another reason is that may be improper transcription, where something gets improperly converted to MA, and the start codon or stop codon is placed prematurely or too late. So the one about polymerase enhancers, or the one about alternative splicing makes different exons, I got the point for. One that I could have added was that introns are removed from the mRNA but remain in the gene structure. The next one that I got wrong. Oh, that's all that I got wrong in this sense. So, so I'm done.